CC92480, which we've christened Mezigdomide, or Mezi for short, which is a little easier, is one of a really exciting new class of oral agents. It's a so-called CELMoD, and really these are actually what I would better define as degraders. They're incredibly powerful engagers of the cereblon E3 ligase complex, and they are a step up from the original parent IMiD. They're a different drug in that sense, actually, because they're synthetically designed to specifically engage with the cereblon E3 ligase complex into a specific pocket in a way that is sort of that much more powerful and that much more potent than, for example, IMiDs do. So these are different drugs. There are two, iberdomide and now Mezigdomide. Mezi is obviously what we're focusing on, and we've shown that Mezigdomide combined with Dexamethasone in relapsed refractory disease, triple class refractory, can engender a response rate around 55%, which is pretty good for an all-oral approach in such heavily pretreated patients. So in this study, we've gone the next step, which is to combine Mezigdomide and Dexamethasone with backbone agents. And our particular focus in our presentation is on Bortezomib exposed patients, or rather, I apologize, on Mezigdomide plus Bortezomib versus Mezigdomide combined with Carfilzomib. So there are two cohorts for the Bortezomib group. One was in a more heavily pretreated group, around 2 to 4 prior lines of therapy. We're very encouraged that we had about a 68% response rate there in this refractory population. Obviously, they had prior Bortezomib, they'd had prior immunomodulation, they'd had prior CD38, so it was a very nice signal to see, and the tolerability profile was really good. In the next cohort, we brought that a little bit earlier, so we were using Mezigdomide with Bortezomib in a less heavily pretreated group of patients, 1 to 3 priors, and the response rate was around 80%, which was very nice. The progression-free and duration of response data are all very consistent. Above all, the tolerability was excellent. So we think we've got a nice combination there for going forward, Mezigdomide combined with Bortezomib and Dex. Now, the data with Carfilzomib is a bit earlier, and it's not quite so mature, but it's really, I think, an important combination platform, because obviously most patients get Bortezomib up front. Therefore, when patients relapse, having a powerful platform combined with Carfilzomib really does make sense. So in that space, we've combined Mezigdomide successfully with the Carfilzomib and really seen a favorable tolerability profile. We've only had one cardiovascular event of note. Otherwise, the drug combination has been well tolerated. There have been some DLTs encountered, which have allowed us to expand the dose cohorts. But overall, we're pleased to see a nice response rate and at the same time, excellent safety profile. So again, the response rates in the 70% type region, which again is, obviously I want to be general about that because the data are so early, so they're likely to mature. So I wouldn't say one is clearly more powerful than the other, because that wouldn't be appropriate. There are different patient populations, but I do think they're showing that the consistency of combining a proteasome inhibitor with Mezigdomide is bearing fruit, and there's clear synergy. What's kind of nice about that is it reflects our preclinical data, which suggested that was exactly what we'd see. And what's also particularly nice is in our study, we're enriched for high-risk cytogenetics in all of the cohorts. We're also enriched for extramedullary disease, which is where we think Mezigdomide may have some real benefit, and we're encouraged to see responses in those patients as well.